Good morning, everyone. Good morning. Happy Thursday. We are still about a minute before 10. We'll give everybody a chance to come in. If you would, go ahead and type your name into chat for me, your name or your nickname. And then I will have that as a record that you attended today. Thank you very much. It's great. Still waiting on a couple of folks. Yeah, thank you very much. All right. I hope everybody's week is going smoothly in this strange COVID reality that we're living in. I'll give a little public service announcement. In case you have not heard, you can go to the US Postal Service website now, and you can sign up to get free rapid antigen COVID tests sent to you. Everybody, every household is eligible for 4 tests. So if you go to the USPS, US Postal Service website, you can sign up and they will send you those free rapid antigen tests. Those are nice to have if you need to, say, travel somewhere and you need to be able to test yourself quickly before you go. Some folks like to do a test before they go visit elderly relatives or someone who might be immune compromised. And those tests are now available for us to get through the mail. So just in case you hadn't heard about that yet. The state of New Hampshire, interestingly, also offers to send you a free test, but they are actually sending out PCR tests. They're actually sending out a test kit. You have to spit in a tube, but if you send that back to the state diagnostic lab, they will run a PCR test for you for free. So, so yeah, the testing situation is looking up a little bit right now. All right, very good. I can see just about everybody's here. I think we're only missing one person, and I think I know who that is. So we're going to go ahead and get started. So hopefully you saw the announcement that I sent out to you after class on Tuesday. Do me a favor and put a number 1 in the chat if you read that announcement, if you saw the announcement that I sent out after class. Great. That's great. If you did not see the announcement, what I would like you to do in your notes for today is make a little note for yourself to go into your settings and in Canvas and make sure that you are set up so that when I send an announcement out, you get a notification. Okay. When I send an announcement out through Canvas, that you get a notification. Okay. You can set up Canvas in lots of different ways to make your life easier. So you just might want to check that out today. And of course, the announcement was about finishing up that first lecture that we did on Tuesday by viewing the rest of it on our playlist on YouTube, Introduction to the Microbes. So I don't know if you all have had a chance to do that yet, but that's something you'll want to get done this week so that you can complete your first lecture homework. The lecture homework is due by Sunday at midnight. That's typically when I make lecture and lab homeworks due, on Sundays by midnight. Okay, so that hopefully gives you plenty of time in your schedule to figure out a time to sit down and take that little quiz, that little homework assignment. All right, so our job for today is to have a little virtual lab exercise. And what we're going to be talking about is biosafety, and specifically biosafety in the microbiology lab. There are good laboratory safety practices that are followed in all laboratories, regardless of what kind of work gets done. But there are some particular safety issues that we have to be aware of when we're working in a microbiology lab. And that's because we're working with microbes. And while the vast majority of microorganisms are not harmful to us, remember, somewhere between 1 and 5%, 1 to 5% of microbes are capable of causing disease in humans and other animals and plants. The rest of them are either actually helpful to us or don't bother us at all, aren't helpful or harmful to us. So while there are very few microbial pathogens that we need to be worried about or protect ourselves from, we do have to protect ourselves from them. So we take a lot of very particular practices within the microbiology lab in order to keep ourselves safe. So what I'm going to do now, I'm going to pull up our Canvas page for just a sec. Let me clean this up. There we go. So what you should be seeing on your screen right now is the Canvas modules page. I just clicked on modules over here in the left-hand menu, and I'm going to scroll down to week 1. Remember, this semester in this course, in each week, there's a to-do list. Be sure you click on that list at the start of every week, because it's going to lay out for you in a quick summary exactly what we're going to be doing and exactly what you are responsible for finishing or completing, homeworks and things. So be sure that you click on these to-do lists at the start of each week. So if you go down to Thursday here, it says we're going to go through a lab exercise. The slides are available on Canvas. Be sure to take notes for yourself. Okay. Now, if I go back into modules and scroll down, here's that page that has the materials that we'll be using today. So if I click on that, you can see here's the slides that I'm going to be going through, biosafety in the microbiology lab. And here's a handout for you called Care and Use of the Compound Light Microscope. So we're going to begin today with the slides, with the biosafety. And then if we have time, we'll go through actually a video about the care and use of the microscope. Since we're going to be in class starting next week, since we're going to be in person, we won't worry too much about the care and use of the microscope yet. I do want you to have that handout, though. I want you to have it with you all semester long, because not only does it go through all of the names of the parts of the microscope and what those parts do, but it also goes through, I think, a 5-step procedure on how to focus a microscope onto a specimen on a microscope slide. And that's a process that I want you to learn. I want you to do the exact same thing every single time that you look at a microscope slide. The sooner you learn the steps of how to focus a microscope, the better you'll get at it. If you do the exact same procedure each time, you will have a much easier time focusing on these microscopic specimens. All right, I'm going to share my screen again, and I'm going to pull up the slides that we're using today. Biosafety in the microbiology laboratory. So be sure to take down some notes for yourself. Remember, you have these slides, so you don't need to write down every single thing on all of these slides. Okay, be kind to yourself. All right. So as I said a minute ago, a lot of the safety protocols that we follow in the microbiology lab are the same protocols that are followed in all laboratories. There are certain safety procedures that we follow in every single laboratory, and that's to keep everyone as safe as possible. An example of that are the door signs that you find on laboratory spaces. So as you walk around Keene State, or as you walk around a hospital or any other kind of medical facility or research facility, on the door or next to the door in laboratory spaces, you're going to find one of these signs. And you may have seen these before. We refer to this as a diamond. It's an NFPA diamond. That's the organization that came up with this system to alert people to the hazards of going into that laboratory space. You can see there are 4 colored blocks in this diamond. The red block provides us information about fire hazards. The yellow block provides us information about some of the chemicals and whether or not they are particularly unstable and dangerous. The white box is for what we call specific hazards. There's only a few chemicals that are noted in this box. And then the blue box refers to health hazards. So are there any things going on in the laboratory that could be possibly a risk to your health? Now, the way the diamond is used is by putting numbers in each one of these spaces. And those numbers range from 1 to 4, with 1 being the lowest level of hazard and 4 being the highest level of hazard. So for example, if you saw the number 4 in this red box on the doorway leading into a laboratory, that would signal to you that there are things being done in that laboratory that are high fire hazards. There may be chemicals that are particularly prone to catching on fire in that laboratory. And this box is warning you about that. So you're prepared when you go inside. If you saw number 4 in the blue box, you know that there are chemicals being used or things going on in that laboratory that are potentially quite hazardous to your health. And again, you would want to know that so that you could wear the correct PPE or otherwise protect yourself while you're in that laboratory. This NFPA diamond is a universal system in laboratory safety. So it doesn't matter if you go to a Keene State laboratory or a Cheshire Medical Center laboratory, or if you were to go into a private industrial laboratory. These diamonds are universal. So you can read them the exact same way, regardless where you go. And placing this diamond on the doorway to the lab or next to the doorway to the lab is actually required. It's a required safety practice for all laboratories. And if the laboratory does not post an NFPA diamond, they can actually get in trouble with governmental agencies who come and inspect labs periodically. So this is information that is provided to you so that you know what kind of hazards you're facing if you're either working in that laboratory or if you're just coming in and out of the lab. In diagnostic laboratories particularly, there are lots of people sort of coming in and out of that space all day long. And the NFPA Diamond allows them to know, what are they walking into? What kind of a space are they walking into, so that they can take the appropriate measures to protect themselves. Now, something else that is universal across all laboratory spaces is this: we don't eat or drink in laboratory spaces. Now, sometimes, particularly in educational laboratories, this rule is not exactly strictly enforced, but we will enforce it in our laboratory this semester because it is a universal safety practice. We do not eat or drink in laboratory spaces. And hopefully that's an obvious one to you, especially if we're in a microbiology lab and we're using living organisms in our work. We would not want to run the risk of eating or drinking anything that could potentially become contaminated with microbes. And something related to that that might not dawn on you right away is this next one. You don't want to do anything like apply makeup in a laboratory or put lip balm on or lipstick on. You don't want to be doing things that potentially expose you to contaminants. Now, if you're putting makeup on, if you're pulling out a small mirror and adjusting your mascara, for example, you're running the risk that there could be microbes on your mascara wand now because you're in a micro lab and you are literally putting those microbes onto your eyelashes or onto your lips. So we just don't do these kinds of personal things in a working laboratory. We also try not to handle our eyes, our nose, our mouth unnecessarily. I tend to have a habit of touching my eyes. I think it's probably because I wear glasses. I have a habit of just touching my eyes during the day. And I have to remind myself when I'm working in the laboratory to keep my hands away from my face. Your eyes, your nose, and your mouth are common entry points for microbes that could cause you harm. As we all know through the pandemic, covering our nose and our mouth, we have to be aware of this. We have to be aware of the fact that there are places on our body where microbes can gain access. And while most microbes are not going to harm us, there are some that might. So we try just not to. We try not to handle your eyes, handle your nose, handle your mouth while you're working in the laboratory. Next on the list is using gloves correctly. Gloves are just about the one piece of personal protective equipment, or PPE, that you're going to see used the most in laboratory spaces. Obviously, there's lots of different kinds of PPE, masks, as I mentioned, certain types of laboratory coats or scrubs that people will wear in a lab space, safety glasses in some lab spaces. But gloves are certainly the most common. We tend to wear gloves when we're working in the laboratory. It's important that we use gloves correctly. And by that, I mean, we put them on correctly, we take them off correctly, and we dispose of them correctly. And yes, there are right ways and wrong ways to do that. We want to make sure that all of the waste that we generate in the laboratory is handled appropriately. Now, this is a particular issue in microbiology laboratories because, again, we're working with living organisms. So we have to make sure that the waste that we generate is disposed of in the right place. Some of the waste we generate just goes in the regular trash. A lot of the things that we use during our experiments and our work each day are just going to go in the trash because they're not a threat to anyone. But there are other materials that we handle that come in contact with living cultures. And that word culture means something to us in microbiology. a culture is a container that is providing a space for microbes to grow. So we purposefully grow microbes in a culture. And if we're using a culture or handling a culture, we are taking the microbes out of there and putting them on things, putting them on what we call inoculating loops, putting them on into test tubes, putting them onto Petri dishes. Anything that comes in contact with living culture has to be disposed of in a particular way. It has to be first decontaminated. And the way we decontaminate in the microbiology lab is by sterilizing. So all of the waste that we generate that comes in contact with our living cultures goes into an autoclave bag. It goes into a bag that will then be placed into an autoclave. Now, if you're unfamiliar with autoclaving as a process, we will be talking about it in more detail. But for now, understand that an autoclave is a machine that uses heat and pressure to sterilize, to kill all of the microbes that might be present. So anything that touches live culture in the laboratory goes into a bag, an autoclave bag, and that bag then gets placed into the autoclave in order to kill all of the microbes. We do not want to put microbes into the regular trash because someone might get exposed to those microbes. And we would not want that to happen because, again, it's potentially dangerous to them. The other thing we have to pay close attention to is where we put our chemicals. The number one chemicals that we use in the microbiology lab are culture materials, the kinds of broths or liquids that we use to grow microbes, and the type of agar or solid media that we use to grow microbes. We want to make sure that that gets disposed of correctly. And we also use a lot of stain. We use a lot of colored stains in the micro lab because we stain these microbial organisms so that we can see them better under the microscope. All of our stains are hazardous waste. And that means they do not go down the drain. We're going to be using stains this semester. We're going to learn some common staining protocols. And when we stain cells that are placed on a microscope slide, when we stain them, we generate waste. There will be excess stain that we have to collect and throw away. We don't put it down the drain ever. Instead, we put it into what are called waste bottles, hazardous waste bottles. And when those bottles get filled up with waste stain, the environmental protection people at Keene State College will come and collect them, and we'll make sure that they are disposed of properly so they don't end up in the landfill. Last on our list here for sort of general things is about washing your hands. Hopefully we've all become like experts at washing our hands during the pandemic. Everyone is washing their hands more than they probably ever have. The general practice in the laboratory, particularly the micro laboratory, is to wash your hands at the beginning of the day and wash your hands at the end of the day. We come into the laboratory and we wash our hands before we begin our work. And at the end of the day, when we're all done and we're ready to go home, we wash our hands before we leave. It's just good practice. One of the things you're going to hear me say again and again this semester is that there are two key things, two key practices in microbiology. Number one, we don't want to contaminate our work with microbes that are present on us. And number two, We don't want to contaminate us with microbes that are present in our work. So a lot of what we're doing is to try to prevent those two things from happening. Either our important work getting contaminated with our microbes, or our bodies getting contaminated with the work we're doing. So by coming into the lab and washing our hands, we're making sure that we're cleaning off any organisms that might be on our hands that might contaminate our work. And at the end of the day, washing our hands before we go home, we're making sure that we're not carrying any microbes out of the laboratory with us on our hands. So when we arrive to class or to a workspace at a job that involves work in a micro lab, there's a couple of things that we do every single time we come in. And this is what I want you to practice this semester while we are in our micro lab at Keene State. Number one, like we said, you want to wash your hands when you get into the lab. And then you want to make sure that all your personal items are stored away somewhere. Now, when we're in the TDS laboratory, in the micro lab in the TDS building, we use a space that's up by the door to store backpacks and coats and sweaters and whatever else people might have with them. The idea is you want to keep your personal items separate from your workspace, again, to decrease the chances that you're going to contaminate your personal items and carry microbes out of the lab with you. Once we have a nice, clear, clean, open workspace, we need to disinfect it. Always, first thing. And there are two disinfectants that we use very commonly in microlabs. Number one is an alcohol solution, and number two is a bleach solution. Now, if you're going to use alcohol as your disinfectant, you can use just about any alcohol, isopropanol, ethanol, plain old ethanol. There's lots of alcohols that are used to disinfect. But here's the key. We don't use a straight alcohol solution. We use a diluted alcohol, 60 to 90% alcohol. One of the things that we've learned during the pandemic is that the general public does not have a good understanding of this. The general public wants to think that if 60 to 90% alcohol is a good disinfectant, then 100% alcohol must be a better disinfectant. I think that's human nature. People think if this littler amount is good, then a larger amount must be better. And when it comes to disinfecting with alcohol, that's just not true. It turns out that a diluted alcohol solution, again, 60 to 90%, is better at disinfecting than a 100% solution. The addition of the water actually makes any microbial organisms more susceptible to the alcohol. So typically what you'll see, at least in my experience, is a 70% solution. Most of the labs that I've worked in use 70% alcohol. So that means there's 70% or 7 parts of alcohol and 3 parts of water in the bottle. We spray it all over our work surface, and then we wipe it off and let it dry. Now, the other very commonly used disinfectant is bleach. Now, I personally don't like working with bleach, even though we use a very dilute solution. We use 10%, a 10% solution of bleach as our disinfectant. That means 1 part bleach, 9 parts of water. And that is the best solution, the best percentage to kill microbes. But here's why I don't like it. I don't like the smell of bleach, number one. And personally, I always manage to get that spray bottle on my clothing. And as you know, bleach will ruin your clothes. Bleach will remove color from clothing. So my personal preference is alcohol. I like an alcohol disinfectant. And that's what we tend to use in our micro labs at Keene State. So we disinfect our work surface and we're good to get started. Next thing we do is gather everything we're going to be using that day, and we gather it all at once. And the reason we do that is because the last thing that you want to happen is to get started on an experiment, get started on a protocol, get your gloves on and any other PPE you might need, have your experiment going in front of you, and then realize you don't have something that you need. That's going to force you to take your gloves off and go hunting for whatever you need. And it's just going to interfere with your work. So we always gather all of our supplies. We also always make sure that we know exactly what's in front of us. And by that, I mean, if you're using a whole range of chemicals that day, if you're using 6 different stains, because you're going to do different staining procedures, it's important that you know which one is which. It's important that you know if you have both a buffer chemical in front of you and a salt chemical in front of you, that you know which one is which. So you don't accidentally put the wrong chemical into your culture or into your container that you're working with. For that reason, one of the other things we are very careful about in the microlab is labeling. We're very, very careful about labeling all of our containers. Now, that's good practice in any lab. That's good practice in a biology lab or a chemistry lab. It's good practice in any laboratory that you label containers correctly. So I've given us a little list here that I want you to learn and practice this semester, which is the way we label containers that have microbes in them. Of course, you're going to want to put your name on that container that you make, either your name or your initials. Most people just put their initials down. You're always going to want to put a date on that container, the date that you produced it. So whether I'm making an agar plate, like a Petri dish plate, or I'm using a test tube to keep my microbes in, I'm going to want to label that container with my initials, with the date. You're going to put down the name of the microbe. What's in there? What microbe is in that tube? And we use the scientific name. And finally, you're going to want to jot down what kind of culture medium is in there. What exactly is the fluid or that sort of gel-like agar material that's in that container with those microbes? Now, you might think to yourself, I don't need to do that. It's my container. I'm the one who made it, so I know what's in it. But here's the thing. There are no microbiology labs that are run by a single person. There are no diagnostic labs in hospitals where only one person works. And since when we use microbes, when we grow them in culture, we always keep them around for a little while. It's very important that anyone who picks up that container can know exactly what's in it. Microbes like nice, warm, dark environments. So we use incubators a lot in the microbiology lab. And we often leave culture containers in incubators for 24 to 48 hours. It's important that anyone who reaches into that incubator and grabs a container knows exactly what's in that container. You would never want to put an unlabeled container into an incubator. It's just not safe. Something else that we talk about a lot is what we call lab hygiene. This is a great way to describe this. It's just the general way you present yourself in the laboratory. And again, this is the kind of thing that applies in all laboratories. but we will also use it in our micro laboratory. Good lab hygiene means you just keep a nice clean workspace. Like we said, you keep your personal things out of your workspace. You define your workspace. In other words, make it clear where you're working on your desktop. Always wear appropriate clothing when you're coming into a laboratory. Now, what do I mean by that? In our lab at Keene State, we don't use any fire, but in a lot of laboratories, Bunsen burners are in use. And you all have experienced the Bunsen burner in chemistry lab and so on. Open flame is one of the biggest dangers in the laboratory space. So one thing that we don't do is we don't come into the laboratory wearing long, flowy clothing. We don't come into the lab wearing a shirt that has a big flowing open sleeve, for example. It's just not an appropriate choice. If you're moving your arm across your desktop and your sleeve is hanging down and touching the desktop, that's not good. Number one, like I said, it could catch fire if a Bunsen burner is in use, but it could also get contaminated with microbes. It might contaminate your experiment. So you want to make sure that the clothing you're wearing is relatively close to your body and not hanging off of you in lots of folds and pleats and things like that. Similarly, you don't want to wear jewelry that's going to hang down while you're working and potentially touch your desktop, your workspace. You want to limit bracelets that hang off of your wrist or necklaces that might hang over and drag across your work. You just want to think about those things. Now you'll notice I wear rings. I wear a lot of rings. And rings are not really a great choice in the laboratory because rings are hard with gloves, with rubber gloves. Now, there is a little trick you can use. For example, if you're wearing a wedding ring and you don't want to take it off, or an engagement ring, there is a little trick you can use. You might even know this trick. The part of the ring that juts up, you just roll it. You roll it over to the back of your finger. That will make it much easier to get your gloves on and off. But many people will just take their rings off. If they're going to be working in the lab, they just take their rings off, put them in a pocket, put them in a backpack. So they're not going to interfere with you getting your gloves on and off. It's important that we wear appropriate footwear in the laboratory. It's not appropriate to wear open-toed shoes in a lab. It's just not. It's not safe. We don't wear flip-flops in a lab. We don't wear sandals in a lab. You just put a good shoe on, a sneaker or some kind of a loafer that's going to cover your foot and make it easy for you to walk around. Okay, so yeah, you wouldn't wear like stiletto heels in your laboratory. It's just not considered good hygiene, good lab hygiene. Something that I have to think about all the time is my hair. If you've got long hair like I do, you've got to manage your hair in the laboratory. And by that, I mean, pull it back. All right, you just pull your hair back and put some kind of a hair tie or rubber band around your hair. It's just the safest thing to do. Hair is particularly vulnerable to catching on fire. Every year, if you look into the records of agencies that investigate lab accidents, there is at least one story every single year of lab personnel setting their hair on fire. And let me tell you, if that happens, it's an awful thing. Number one, it's going to destroy your hair. Number two, it often leads to burns on the skin. And there have been people who have set their hair on fire and then set their clothing on fire. So again, the easiest thing to do if you've got long hair is just pull it back and put an elastic around it. That's going to protect you from open flame. It's also going to keep your hair out of your work. It sounds gross, but it's very easy for your hair to swing forward and just get into your work, get into your culture, get into onto your microscope slides, and it's going to contaminate what you're doing. It's also going to contaminate your hair. So we try not to wear our hair long and flowing in the laboratory. Respect fire. I can't say that enough. Respect fire. It sounds so basic and simple, but like I said, there are stories every single year about people working in laboratories who ought to know better, who don't respect it, who accidentally set themselves ablaze, and who suffer the consequences. There was a terrible story out of the University of California a few years ago now, UC Davis. A very young but very smart graduate student was working in a lab. She was working on an experiment with one of those chemicals that is highly reactive. She had the chemical in a hood, in a chemical hood, so that that chemical could be kept safe and prevent it from catching on fire. She got distracted. She brought her beaker out from under the hood. And when it got exposed to the air, it caught fire. It was a very, very reactive chemical. The fire in the beaker quickly spread to her clothing and her hair. And she panicked. She panicked. And unfortunately, the people in the room with her also panicked. And despite the fact that there were safety features in that laboratory, including a shower, an emergency shower, no one got her over there and turned the shower on. And by the time the ambulance arrived, she was burned over about 75% of her body, and she didn't survive. Respect fire, respect fire. Know how to protect yourself, and know in the laboratory where the safety things are. Know where the emergency shower is. Know where the eye wash is, so that in a moment of an accident, you don't even really have to think, because you already know, oh, the shower's over there. I got to go over there and pull the cord so that I can put the fire out. That story always stays with me, because that young woman was an experienced scientist. I mean, she had gone through her undergraduate, she was in her graduate program. She had worked in a laboratory for many years, but she did everything exactly wrong in that moment, and it cost her her life. Now, like I said, in our class, we actually don't use flame anymore. We don't use Bunsen burners. Instead, we're going to use a device called an incinerator. It sounds terrible, but it's just a little device that has a chamber in it that gets very hot. And when we need to sterilize an instrument, we'll simply put it into the incinerator chamber and let it warm up in there. So we won't be using fire in our lab, but you will probably encounter Bunsen burners in other laboratories, and just always respect them. Be very safe. PPE in laboratories varies tremendously. It all depends on where you're working and what their particular procedures are. So there is, as I said, sort of a universal practice of wearing gloves when you're working in any kind of laboratory. But in some labs, you'll wear a lab coat. In some labs, you'll wear goggles or safety glasses. You might wear a full face mask. It all depends. When we're working with microbes, it depends on how dangerous those microbes are, what level of PPE that we will be using. Now, we will always, always, always use gloves, and in any place that handles microbes, whether the microbes are perfectly harmless or very, very dangerous, always, always, always wear gloves. You have to put them on and take them off correctly. We call that donning and doffing, putting the gloves on and off correctly. Once you have your gloves on, it's important to remember you don't want to touch anything that is not routinely disinfected. Now, what do I mean by that? When you put a pair of gloves on, as soon as they're on, you want to act as though they're contaminated. All right, we put our gloves on and then we behave as though they're contaminated. So I would not take a gloved hand and do this or this. I would not touch a doorknob with a gloved hand on because doorknobs are not routinely disinfected. I wouldn't want to handle my microscope with my gloves on. I would take my gloves off if I wanted to work on my microscope because we don't routinely disinfect microscopes. Okay, so I can touch my, workspace if that's appropriate, or I could touch some of the tools I'm using if that's appropriate, because those things do get disinfected routinely. But when you have your gloves on, I want you to be thinking in your head, my hands are dirty. My hands are contaminated and dirty right now. So I'm not going to touch anything that can't be disinfected at the end of my workday. Now, the other thing about gloves, we don't reuse gloves. You don't take off a pair of gloves and set it down and then plan to put them back on. You always get a fresh pair. So if you need to take your gloves off, let's say, for example, you want to touch something that's not routinely disinfected. Let's say you're working and you need to touch your cell phone, or you've got an itch and you need to scratch it. You would take your gloves off, scratch whatever needs to be scratched, and then get a fresh pair. Don't try to put an old glove on. It's not good practice. Gloves are not expensive. So when you take it off, it's off. It's trash. Now, gloves actually do go into regular trash in the laboratory. Even if they do actually have contaminating microbes on them, those microbes can't survive on a glove for longer than a few minutes. It's the chemical, it's the molecules that are in gloves that prevent microbes from surviving very long. So when you finish working and you take your gloves off, those go into the regular trash. Know where your safety equipment is. In every laboratory, find the eye wash station, know where it is. Know where the emergency shower is. Know where the first aid kit is. And if you use fire in your lab, know where the fire extinguisher is. And sometimes there's even what's called a fire blanket. And that is literally just a big, heavy blanket that's made out of non-flammable material that you can literally just cover a fire with, a small fire, and snuff it out. It's your job, if you work in the laboratory, to know where the safety equipment is, so that in the moment of an accident, you don't have to search around looking for it. Don't touch things when you're wearing gloves. Don't touch things that are not routinely disinfected. Don't touch your face. Don't touch your glasses. Don't touch your hair. Don't touch your textbooks. Don't touch your microscope, your cell phone, your laptop. Don't touch door handles. Pretend that your hands are covered in microbes. They may not be. Your gloves may be perfectly clean, but don't touch things. And then you'll be sure that you're not accidentally contaminating things. And when you're done at the end of the day, take them off and put them in regular old trash. I said it a little bit earlier, and I'll say it again. The two most important things to remember, put a star next to this. We don't want to contaminate our work, our cultures, these containers that contain microbes that we're trying to grow. We don't want to contaminate our cultures with the microbes that are on our bodies. And we don't want to contaminate ourselves with our cultures. You could also substitute in a diagnostic sample here. If you were working in a diagnostic lab in a hospital and you were running urinalyses and you were handling urine samples from patients, you don't want to contaminate that patient's urine sample because then they'll get a false result. But you also don't want to contaminate yourself with their urine sample. So you have to take practices and procedures that will prevent that from happening. Remember, 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 you are covered in microbes. It really grosses people out sometimes when you tell them that, but it's true. You are covered in bacteria and archaea and fungi. You are covered in microbes and you are full of microbes. Your whole body is full of them. And the good news is they're good for you. They protect you. They keep you healthy. They help you stay nice and healthy and strong. There's a tiny percentage of them that are pathogens. So we have to act as though all of them are pathogens because we want to protect ourselves from the pathogens. But of course, the vast majority of them are perfectly harmless and are even helpful to us. They're actually good for us. The human body is covered in microbes. The environment is full of microbes. There are many, many, many more microbial organisms out there in the world than there are humans. When we create a culture in the laboratory, what we call a pure culture, it's got one microbe growing in it. We have created a container that has millions of microbial cells in it. Okay. And if you're handling any kind of material from a patient, a tissue sample, a fluid sample, blood, urine, it doesn't matter. You have to assume that it has microbes in it. You have to assume that it has microbes in it, and then you'll be safe. You would never pick up a little cup full of urine unless you had gloves on. You should never pick up a red top blood tube unless you have gloves on. And you should never pick up a petri dish with a bacterial culture growing in it unless you have gloves on. Even if you know that what you're growing is not a pathogen, you pretend that you're handling a pathogen. And that way, if you ever actually encounter a pathogen, you're practicing the safest methods that you can. In our class, we're not going to handle any pathogens. We're going to handle organisms that are known to be safe. But I want you to pretend that you are handling a dangerous pathogen. Because if you get in the habit of behaving as though every microbe is a pathogen, you'll be as safe as you can be. And that way, if you ever do handle a pathogen, you'll be safe. Does anybody have any questions? Does anybody have any questions about anything I've been talking about before we move on to the next part? Hopefully you've talked about some of this in other classes. Hopefully you've talked a little bit about some of this, but I'm sure some of this is new to you. You have to practice these things. You have to practice these procedures in order to get comfortable with them. By the end of this semester, I want you to feel abnormal if you're going to pick up a container and you don't have gloves on. I want that to feel bizarre to you, as though you would never, ever do it. I want it to become like a second nature to you, that when you're working, you always have gloves on, unless of course you're, working on the microscope or something. But if you're handling a Petri dish or you're handling a test tube, I want it to feel strange if you tried to pick it up without a glove on. It needs to become second nature when you're working in a microbiology lab. The next thing we're going to talk about are what are called biosafety levels, or BSLs. Now, when it comes to microbiology laboratories, there is another regulatory aspect of letting people know how risky that laboratory is to their safety. We talked about the Diamond, and remember that's fire and reactivity of chemicals and general health hazards. But there's another level of sort of safety organization that we apply only to laboratories who are handling potential pathogens. And that's called the BSL, or the biosafety level. So, for example, laboratories that handle the safest microbes, like teaching laboratories, those get a BSL level 1. So if a laboratory is designated BSL1, that means that there are no organisms being handled in that laboratory that are dangerous to us. There are no pathogenic microbes being handled in that laboratory. It also means that the people working in that lab only need to use very basic biosafety techniques, because like the ones we've been talking about, because the risk to them of getting sick is very, very low. So let me pull up our next slide. In a BSL1 laboratory, and again, teaching labs have this designation, we follow what we call standard laboratory safety practices, like we've been talking about today. The PPE will vary a little bit depending on the institution, depending on the people who run the laboratory. But generally, it's things like gloves, always gloves, and maybe lab coats, maybe safety glasses. It depends. It depends on the particular laboratory. Now, when we go up to a BSL2, biosafety level 2, what's going to happen in that lab is they're going to follow all the BSL1 procedures. So again, standard laboratory safety procedures. But they're also going to do this. They're going to limit access to the lab. They're going to limit who can come in and come out. Maybe you have to use a badge to get into that lab. Maybe there's a key code that you have to enter to come into that lab space. Only people who have permission can come in. In that lab, you're also going to see the use of biological hoods. You're going to see a particular form of sharps management, waste decontamination procedures that are a little more involved, and more PPE. Now, in a BSL2 laboratory, they are allowed to work with pathogens, but they're only allowed to work with non-respiratory pathogens. And they have to be pathogens that cause a disease that either has a treatment available or has a vaccine available. So what do I mean by that? Well, a BSL2 lab could, for example, work with polio, or could work with streptococcus that causes infections in a human, or could work with chickenpox or something like that. Those are pathogens that don't cause a respiratory disease, but some other kind of disease. And those diseases have either a treatment available or a vaccine available. So all the people who work in that lab that say handles chickenpox would either have had chickenpox or would have been vaccinated against chickenpox. All the people that are working in that lab that handles that streptococcus, that pathogenic streptococcus, we know that there's an antibiotic available that can treat them if they accidentally get infected. We actually have a couple of BSL-2 laboratories at Keene State. We have a BSL-2 laboratory that handles, I want to say, Vibrio. There's a kind of bacterium called Vibrio that can cause a couple of different types of disease, including intestinal diseases. We have another laboratory at Keene State that handles other microbes that are considered BSL-2 level. So not just anybody can walk into those labs. You have to have a reason for going in there. And the safety procedures are a little more involved. Now, the third BSL level, BSL-3, follows all of the BSL-2 laws and restrictions, but also has, again, controlled access, more extensive PPE, more extensive specialized decontamination procedures for the people who work there. They are allowed to handle respiratory pathogens, but there has to be a treatment or a vaccine available. Okay, so again, a higher level of risk here because they're handling known human respiratory pathogens, but there has to be a treatment or a vaccine available. So a BSL-3 lab might handle influenza. A BSL-3 lab might handle another organism that causes a pneumonia or something like that. BSL-3 laboratories are less common. You have to have a lot of security in place in order to be at a BSL-3 level. We have BSL-3 labs in New Hampshire, but not at Keene State. Now, the final BSL level is called BSL-4, and BSL-4 is the highest level of security because it's also the highest level of risk. In a BSL-4 laboratory, they follow BSL-3 procedures, plus the lab has to be located in a completely separate building from the rest of the facility. They have to wear full body PPE. They have to wear a suit that has its own air supply, a positive pressure air supply. And that means that they're breathing essentially from an oxygen tank. They're not breathing room air. They have to go through a decontamination process when they exit the lab. These laboratories are handling novel pathogens. In other words, brand new pathogens that we don't know much about. They're handling respiratory pathogens. And these are pathogens that have no treatment yet, and there is no vaccine available. So yes, SARS-CoV-2, the pandemic virus, back in 2020, was only being studied in BSL-4 laboratories. Now, once a vaccine became available, that opened up the opportunity for more laboratories to study that virus, because now people in BSL-3 laboratories could study that virus, now that there was a vaccine available. There are very few BSL-4 laboratories in our country, but there are actually a couple of them right in Massachusetts. We have a couple of BSL-4 labs right in Massachusetts. It is a high risk laboratory. It has very high safety procedures, and you better believe it's very secure. You have to pass through several security points before you can even get near a BSL-4 lab. And very few people work in those labs. You have to be specially trained to work in those labs because of all those safety procedures. In this slide over here on the left, you're looking at a person who's working in a laboratory that is BSL 1. So as you can see, this person has some kind of a specialized clothing on. It's some kind of scrubs or something that she has on, but otherwise she's not wearing any particularly extensive PPE. She has her gloves on. She's working over here. She's got her hair pulled back. Good. We like to see that. This is just some research laboratory where she's not handling any kind of pathogenic organisms. She's not handling any microbes that could harm her if she were to bring them into her body somehow. Now, if you compare that to this picture, this person is working in what we call a biological hood. And this you would find particularly in BSL-2 laboratories. So you can see her arms are underneath this glass door that you can pull down. And most of the work she's doing is inside this cabinet. Now that cabinet looks a lot like what we call a chemical fume hood. You've probably seen lots of chemical fume hoods if you've ever been in a chemistry lab. A fume hood, like the name suggests, literally sucks all the air out of that workspace so that you don't get exposed to any chemical fumes coming out. All right, that's a very different type of hood than a biological hood. What a biological hood does is circulate air in a way that prevents it from coming out and also prevents the surface, the work surface in there, from getting any microbes on it. So it's a sterile workspace. A biological hood is a really nice thing to work in if you're ever working with something that you have to keep very sterile. And those are found in BSL-2 labs especially. We have a couple of biological hoods here at Keene State that we sometimes use in coursework. Now, there is one problem in this picture. Type into chat if you can see what this young woman is doing wrong here. She's got a nice lab coat on. You can't really see it, but we're going to assume she has gloves on. What is she doing wrong? Type it into chat if you think you know. Yeah, you got it. Everybody's got it. Oh my gosh, her hair is hanging down. Oh, that'll send me into a fit if I see that. Yeah, that's not good practice. That's not a safe practice. She should have her hair back in a ponytail. Now on this slide over here on the left, you're looking at a person who's working in a BSL-3 laboratory. Notice how much PPE she's got on. She's got a surgical mask on. She's got a gown on, her gloves. She's got her hair underneath one of these surgical hats. She's working in a biological cabinet. She's doing all her work in here in this sterile environment. Okay, this is BSL-3. Over here, this person is working in a BSL-4 lab. How do I know? The suit. This person is also working in a biological hood, but notice the suit. This is a self-contained environment. All right, remember, what this person is handling is a respiratory pathogen. It is potentially a brand new pathogen, like we saw with SARS-CoV-2. It has its own air supply. We cannot allow this person to breathe room air because this pathogen may have gotten out. It may be out in the air, and we want to keep this person as safe as possible. So this red hose right here is feeding oxygen into her suit. She is completely isolated in that suit. And when she has finished working, she's going to have to go through a decontamination shower that's going to wash any potential pathogens off of that suit before she takes it off. It's not easy to work in a BSL-4 lab. You spend most of your day just getting ready to go into the lab, and then you spend another hour coming back out of the laboratory. So it's not easy. It's not an easy place to work. Now, when it comes to these BSL levels, I don't want you to worry about learning all of the restrictions in each lab. What I'd like you to know is that a BSL-1 lab is the lowest level of hazard, and a BSL-4 lab is the highest level of hazard. That's the most important thing to know. And know that only some laboratories are allowed to work with dangerous pathogens. And that's because we have to take a lot of procedures to keep the workers safe and to keep the community safe. We don't want to allow dangerous pathogens to escape the laboratory and come out into the community. Does anybody have any questions about the BSL levels? There are a couple of great movies out there that are about, pandemics, essentially. And some of them actually do a good job at showing what happens in these laboratories. Some of them don't do as good a job. They make up some things, but there's a great movie called Outbreak that's about a pandemic, or at least an epidemic. I can't remember. And there's a scene in that movie where people in a BSL-4 lab have an accident and somebody's suit gets breached. Somebody's suit gets, torn, and they get exposed to the pathogen. Again, those kind of things happen very, very rarely in a real laboratory, but it makes for a good movie, makes for a good story. All right, we have a couple more slides to look at. Couple more important things to remember. Always assume when you go into the laboratory that your workspace is contaminated. All right, always assume that. Now, we're going to come in, we're going to disinfect, right? We're going to disinfect our work surface before we get to work. But as soon as we finish disinfecting it, there's a potential that it gets contaminated. So we always act as though it's contaminated. Now, what does that mean? It means that you would never place uncontaminated equipment, sterile equipment, down onto your work surface. As soon as you take a sterile tool out of its package and you place it down onto your workbench, you have potentially contaminated it. It also means that you would never take a contaminated piece of equipment. Let's say a pipette that you just put into a culture, a liquid culture. You would never put that down onto your workspace because you're now adding more microbes onto your workspace and contaminating it. We handle inoculating loops a lot in this course, and we'll be talking about them in a little bit. It's the most handled piece of microbiology equipment because it's the tool that we use to move microbes from one place to another. Those have to be sterilized again and again and again, using those incinerators that I mentioned. We have to make sure that we never take a contaminated inoculating loop and put it down on our workbench. We would also never take a sterilized inoculating loop and put it down on our workbench. All right, if it's sterile, you've got to keep it sterile, and that means keep it in your hand. Caps, lids, syringes, needles, petri dishes, all those things. If they're sterile, you've got to keep them sterile, and you cannot place them down on your workbench without them becoming potentially contaminated. We're going to decontaminate our inoculating loops before we use them and immediately after we use them. And remember, in some labs, that's going to be done with a Bunsen burner. In our lab, it's going to be done with an incinerator device. And in some labs, you'll even see alcohol lamps used. If you've never seen an alcohol lamp, it's literally just a little glass dish. It has a wick coming out of it. You fill the dish with alcohol and you set the wick on fire. It's kind of like the old-fashioned lamps that people used to use in their homes. Obviously, it's an open flame. It's a little bit dangerous, but you do see them from time to time in laboratories. We handle culture containers in the microbiology lab. We handle Petri dishes that have microbes growing in them, and we handle test tubes that have microbes growing in them. We always wear gloves when we touch culture containers. We always keep the test tubes in a rack so that they're standing upright, because we don't want to spill a liquid that's full of microbes. Here's something else, and we'll talk about this more as we go through this semester, but Petri dishes that we use to grow microbes, we always keep upside down. Now that seems bizarre. Petri dishes have a lid that goes on them. The microbes grow in the dish itself on that semi-solid sort of gelatinous agar material. And then we put a lid on to keep it, uncontaminated. But we then flip the dish over. The reason we do that, I mentioned incubators earlier. If you put a Petri dish that's growing a culture on it, if you put that into the incubator right side up, there's water in that agar material that the microbes are growing on. And what will happen in the incubator is the water will start to condense on the lid, on the underside of the lid, and it will drip down on the microbes, and it will interfere with them growing. So instead, we always keep it upside down. And that way, the condensation stays on the bottom, and our microbes can grow happily upside down. It's very strange. But again, you'll get used to it in the laboratory as we start to work with Petri dishes. Remember, anything that touches culture, any culture that we're done with, has to go into one of these special bags. This is an autoclave bag. Notice the biohazard symbol on it. When this bag is full with contaminated materials, we're going to tie it off, and we're going to put it in that autoclave in order to sterilize it before we throw it away. The teaching laboratory that we're going to be in this semester is at a BSL-1 level. We don't handle pathogens. Most microbes are not pathogens, but we're going to treat every microbe that we handle as though it's a pathogen, because that way we will develop the best skills that we can possibly develop at keeping ourselves safe. Here's something to remember, and you'll hear me say this several times this semester as well. Contaminating microbes that are on us or in the environment, in the air, for example, they don't fly. Microbes don't fly. In other words, you can't be contaminated by simply opening up a culture. The microbes cannot jump up out of the culture and contaminate you. Microbes fall. They fall out of the air. They fall out of our breath. So we don't keep anything sterile open any longer than we have to. If I were to have a culture dish in my hand or a test tube that had culture in it, and I wanted to keep that culture from getting contaminated, what I would not want to do is open it up and just leave it exposed to the air, because microbes are going to fall out of the air onto my dish or into my tube and contaminate it. So if I have to open a culture, I'm just going to be sure that I only open it as long as I need to, and then I'm going to quickly close it again. I don't have to worry about microbes jumping up off a surface, but I do have to worry about them falling down out of my breath or out of the air. So we always only open our culture containers for as short a period of time as we need to, and then we quickly close them again. All right. So a lot of this stuff we will talk about again and again through the semester while we're working, while we're doing experiments. But it's a good introduction. Does anybody have any questions about anything that I've said today? Any questions about any of the safety procedures? Any questions about some of the specific microbiology related procedures? Feel free to type it into chat if you do. Again, we'll talk. You'll hear my voice again and again in the laboratory saying things like, close that container, keep that container closed, don't leave that container open too long, or turn that dish upside down, make sure you turn your dish upside down. Things like that. It just takes practice. It just takes practice. It's like anything else that you're learning. All right, very good. All right. When we get together next week, before we start talking about the microscope, when we get together next week, we're going to talk about what's called aseptic technique. That is a whole group of procedures that we do, techniques that we do in the laboratory in order to, again, prevent contamination. So we'll talk a lot more about this next week when we get back together in person, hooray, in the laboratory. The last thing that I want to do with you today is talk just a little bit about microscopes. And again, it's hard to practice focusing a microscope when you're not actually handling a microscope. So we won't worry about that too much today. But it is important that you learn the pieces and parts of a microscope. So number one, so we can talk to each other intelligently. If I tell you to, for example, adjust the diaphragm or adjust the condenser on a scope, or if I tell you to use the fine adjustment knob versus the coarse adjustment knob, it's important that you know what those things are and where they are. So I've put together a handout for you about what are called compound light microscopes, modern light microscopes. And we're going to handle those microscopes in the laboratory. Compound light microscopes are very complex, high-tech tools for us in the laboratory. And they're not cheap. A nice microscope, a teaching microscope, costs about $3,000 to $4,000. They are precise optical instruments, and they're very useful. And if you're going into any kind of healthcare related field, it's important that you understand, number one, how microscopes work, like what they do, and number two, what the pieces and parts are on a microscope. It's always very, very useful to know how to focus a microscope so you can look at something. So I'm going to pull up that handout for us right now. And this handout is on Canvas for you. And that's what you should be looking at right now. Care and use of the compound light microscope. Now, this word compound, it actually means something to us. It means that we're talking about a microscope that has more than one lens on it. I'm going to scroll down and we're going to look at a picture of this microscope, sort of a general purpose microscope. You'll notice down here, there's 1, 2, 3, 4 different lenses on this scope. And each one of these lenses has a different magnifying power. On a typical compound scope like this, there's going to be a low power lens and then sort of a medium power lens. Then there's going to be a high power lens. Then there's going to be what's called an oil immersion lens. So all compound microscopes have multiple, what are called objective lenses. That lens that's going to allow you to magnify the specimen is called an objective lens. And it has some power associated with it, some magnifying ability associated with it. On a typical compound light microscope, the low power lens, it's going to be either a 4 or a 5x. So it's going to magnify the specimen 4 or 5 times, depending on the lens. The second lens is going to be either a 10 or a 20x. So again, it magnifies either 10 times or 20 times, depending on the scope. Then you're going to have what's called a high dry lens. And that one is 40x. It magnifies 40 times. And it's called high because it has a high magnifying power. It's called dry because you don't have to put anything on your specimen to use this lens. High dry. The final lens on a compound light microscope is called the oil immersion lens. This lens can magnify 100 times. That lens requires that you put oil on top of your slide. That lens will not work unless it is sitting in a bubble of immersion oil. It needs the oil to focus on the specimen. All compound microscopes have multiple objective lenses. And depending on the scope, you'll get those 4 types of lenses. So you've got a diagram here that names these parts. Obviously, there are eyepieces. A modern microscope, light microscope, has 2 eyepieces. And it's really important that you open both eyes when you use these modern microscopes. In the old days, we used to use single eyepieces. We called it monoocular. Mono meaning one, ocular meaning eyepiece. But a modern microscope has 2 eyepieces, so it's called binocular. Bi meaning 2. So we open both our eyes when we look through a modern microscope. There's going to be a tube that comes down from the eyepieces towards those objective lenses. That's called the body or the body tube. Then there's going to be what's called an arm. And this is the part of the scope that we grab onto when we want to move the scope. The place that you set your slide onto, that's called the stage. And the stage is going to have a couple of clips that you can open so that you can put your slide in and sort of secure it with those clips. There are going to be 2 knobs on the side of the scope that are going to help you focus. The larger knob is called the coarse adjustment knob. And the smaller knob is called the fine adjustment knob. The coarse knob just makes bigger movements than the fine knob does. So we start focusing using the coarse knob, and then we do our fine focusing with the fine knob. Now, every scope has a base, and it's going to have some kind of a switch on it that turns the light on and off. All of these scopes have to be plugged in. They need an electrical source to generate that light, and the light is going to come up from the base. The light comes up and it travels through a couple of other pieces of equipment before it hits the specimen. From the specimen, it then goes into the lens and through the body tube and up to your eyes. I got your message, Rebecca, no problem. It's important to note that those magnifications I mentioned for the objective lenses, those are for the lenses themselves. Those 2 eyepieces, they also have lenses in them. Each one of those eyepieces that you look through can magnify 10 times. So that means if you want to figure out the total magnification that you're using to view the specimen, you have to take the number from the objective lens and multiply it by 10 for the eyepiece. So for example, if I'm using a 20x objective lens that magnifies 20 times, I'm really magnifying the specimen 200 times because I'm using a 20x lens and a 10x eyepiece. All eyepieces are 10x, by the way. So 20 times 10 is 200. I have magnified whatever I'm looking at 200 times. Now that means if I use the highest power lens, that 100x lens, the oil immersion lens, if I use that lens, 100 times 10 for the eyepiece means that I'm looking at a specimen magnified 1,000 times. That's the limit. That's the limit of our magnification ability on a typical compound light microscope. Our limit is a 1000x magnification. But we can see a lot with that. We'll be able to see the general size of the bacterial cells. We'll be able to see their shape. And we'll be able to see if they have any appendages, like if they have flagella coming off of them or anything like that. So the compound light microscope is used in every microbiology lab to help us visualize the microbes that we're looking at. And it's important that we know the names of their pieces and parts. Now, there are two other pieces on here that I haven't mentioned yet, and these are kind of confusing for students. Underneath the stage, remember the stage is that flat square that you're going to put your slide on. Underneath is what's called the diaphragm and the condenser. Two rings that are under here that the light is going to pass through before it passes through the slide and goes up into the lens. The diaphragm and the condenser. Now, if you scroll down in the handout just a little bit, the top of the handout just gives general instructions on how you handle the microscope, like when you're taking it out of the cabinet at school. And then, of course, there's this nice image. And then if you scroll underneath the image, there's sort of a checklist for you the first time you use the microscope to make sure that you understand where all the pieces are. Look at this second paragraph. The substage assembly, that's what's right here underneath the stage, includes the diaphragm, and the diaphragm has a lever on it or an arm, the condenser lens, and then what's called a filter holder. The diaphragm is going to change the diameter of the beam of light. The condenser is going to concentrate that beam of light onto the object that you're looking at, the microbe. The filter holder, like the name suggests, can hold a colored light filter. We have the ability with our microscopes to put on these little discs that have different colors, pink and blue and yellow, because sometimes changing the color of the light helps us see things a little clearer under the microscope. Our microscopes that we're using in TDS, I don't think have filter holders, but don't hold me accountable to that. I have to double check. A lot of light microscopes have filter holders, which is nice. It's just another tool we can use. All of the microscopes, though, are going to have that diaphragm and that condenser. Okay, those two pieces are critical. Remember, the diaphragm is going to make the light beam wider or narrower, and the condenser is going to focus it in on our specimen. It's easy to forget that the diaphragm and the condenser are down there. And we have to remember it because sometimes we need to adjust them in order to get the best view possible for our specimen. Now, if you scroll down, there's a couple important things to know about these microscopes, and I have them in this yellow highlighted color. Never, never rotate the 100x objective lens into position over your glass slide unless you have put oil down on your glass slide. If you take that lens and you put it in position and there's no oil on your slide, that lens is designed, it's built in a way that it's going to come in right on top of your slide. It's going to look like it's going to break your slide as you slide it into place. If there's no oil on that slide, you're going to scratch the lens. So before you put a 100x lens in place, you just put a little drop of oil on your slide, and then you put your lens in place. So that's one thing to remember. Here's something else that's important. Objective lenses get dirty. The oil immersion lens, of course, gets oil on it. We put oil on it. So anytime we use that microscope, when we're finished, we have to clean the lenses. And it's very important that you use the right material to clean objective lenses. You have to use lens paper and lens cleaning fluid. Any other cleaner and any other material that you would use to wipe it off, and you will damage the lenses. There is special paper that is just for ocular lenses, and there is a special cleaning fluid that is just for ocular lenses. So it'll be very important to remember that this semester while we're working with the microscopes. I told you that there's a procedure that we're going to learn that helps us focus in on a specimen. I've got that procedure listed here. You can see the steps. There's actually 9 or 10 steps. Okay. Don't worry about that right now, because again, until you have a microscope in front of you and until you're actually trying to focus on something, it's not going to make a lot of sense. So again, what I'm going to do is I'm going to print that handout I'm going to print it out for everyone, and I'm going to distribute them in our lab when we get together next week. And then I'll just have you keep it with your notebook. And every time you are tasked with focusing the microscope on something, you can pull it out and you can look down that list of steps. And if you follow the same steps every time, you will learn them very quickly, and you'll have the best chance of focusing on your specimen with ease. Of course, I'll be in the lab the whole semester to help too, because it does take practice. The hardest part of working with a microscope is learning how to focus on something that is so small on your slide. All right, now I want you to know there is a video available to you. It's on our playlist. It's called Care and Use of the Compound Light Microscope. And it's basically just a video that talks through what we just said, what I just said to you. It talks about the pieces and parts. It talks about how to focus. So that's available to you all semester long if you need to bring it back up and take another look. Okay. We'll get lots of practice with the microscopes this semester. All right. Does anybody have any questions? Does anybody have any questions about microscopes or about the safety procedures that we talked about? Does anybody have any questions about anything we talked about today? Okay. Now remember, you have a lecture homework to do for me by Sunday at midnight. You have to view the rest of that lecture, the rest of the lecture that we didn't get to on Tuesday, before you'll be ready to complete the lecture homework. So if you haven't done that yet, That's the next thing that you'll want to do. Watch the rest of the video. Go to that announcement I send out because I tell you exactly where to go in the video. Watch that, take good notes for yourself, and then you'll be ready to do that first lecture homework. You have a student survey to finish. Most of you have submitted that. Thank you. Thank you very much. If you haven't, that's in the Start Here module. You'll want to finish that survey and get that into me. And then you'll be good for the rest of the week. Okay. And when we get together on Tuesday in the classroom, yay, we're ready to do another lab. Okay. That material for next week's lab is already posted up on Canvas for you. The to-do list is there for next week. The slides we're going to be using, what you're going to need to do next week. It's all posted for you so you can take a look whenever you want to. Okay. But all you have left to do for this week is make sure your survey's done and do that lecture homework for me. I think there's 10 questions. I want to say there's 10 questions, but maybe not that many. Hopefully everybody will get 100% on it. All right, everybody. If there's no other questions, I'm going to let you go. Enjoy the rest of the day. Enjoy the rest of the week. Hopefully it's been a nice, smooth start to the semester. And I will see you again next week. Don't hesitate to message me if you need anything.
Thank you.
You're welcome.
Thank you.
Thank you.
You're welcome, everybody.